guys, how's it going? Today I'm going to tell you a little bit about DNA. DNA is an acronym. Acronyms are when we have just the front letters of words representing an entire long word. And so DNA is an acronym describing deoxyribonucleic acid. OOFDA. That is a big word. And so we shorten it up to be DNA. D means not. Oxy means oxygen. Ribo is a type of sugar. And nucleic acid is a molecule. So the huge word DNA, that sounds really scary, is basically just telling you is that it's a molecule with sugar that doesn't have oxygen, which is kind of strange for a type of sugar. So DNA is deoxyribonucleic acid, a molecule that has sugar without oxygen. All right, so let's talk a little bit about DNA. DNA is in all living things. One of the characteristics of life that makes something living is that it has to have DNA. So if something doesn't have DNA, it won't be considered living at all. DNA has a code within it, and that code makes proteins. And proteins make almost everything inside a human thing or a living thing's body. So DNA has a code which makes proteins, which make us. We as humans are made because of the code inside of DNA, which is really mind-boggling and tough to think about. DNA is located inside the nucleus of a cell, and it can come in two different forms inside of that nucleus. When it's really messy and crazy and stringy all over the place, it's called chromatin. T-I-N. But when it's nice and organized, when that cell's about to divide, it's called a chromosome. S-O-M-E. Now, this messy DNA that's all over the place, or a nice X-shaped chromosomes, have strands of DNA in them. And that strand of DNA have different sections of it. And these sections of DNA are called genes. And these sections or genes in DNA are what make certain things inside of a body. So one section of DNA could be the gene that codes for hair color. And another section could be a gene that codes for eye color. And another few could be a gene that codes for skin color. And so all these different sections have different genes that make certain things. The usable section of DNA is called an exon. The exons are taken out of the DNA and used to make proteins, which make us. But some of these sections of DNA aren't really useful. They're kind of just hanging out and sitting there and taking up space. And those sections of DNA are called introns. So DNA is a long string that can be messy or organized, and that long string has genes on it. And those genes will make proteins, and we're made of proteins. Let's talk a little bit about the shape of DNA. The shape of DNA is oftentimes called a double helix. You'll hear it called a double helix over and over again. When someone's calling DNA a double helix, they're saying it has double two strands. And so we have one strand here, and we have one strand here in the DNA. The strands are made up of two different things. They're made up of these pentagon-shaped guys, and those are called sugars. So these things that look like pentagons are sugars. Also making up the strands are these little circle guys hanging off. And these are called phosphates. The phosphates are what is responsible for binding these ribose sugars together. Now remember, DNA stands for deoxyribose, and that ribose is the sugar. So these sugars right here don't have oxygen, which remember is a little bit weird for a sugar. So overall, we have one strand right here of DNA, and we have another strand right here of DNA. Now that strand isn't just a flat strand like it's shown right here, nice and flat on the board. It's actually making a twisting shape. And so that twisting shape we call a helix. That helix is how it turns and spins. Now DNA doesn't turn both ways, like sometimes it's represented on posters or even textbooks sometimes. If you hold up your right hand and now move your hand up and to the right, that's the direction that that twisting shape in DNA will go. If you were to use your left hand and twist it up, that wouldn't be the direction that DNA would go. And so because of that, DNA is called right-handed. It turns to the right. As it turns to the right, it makes one complete turn around every 10 bases. So every 10 bases, the DNA makes one turn. The bases are going to be these rungs on our ladder. Or if you look over here at this picture of a DNA ladder, each one of these things is going to be called a base. And so for every one turn in the DNA, there's going to be 10 bases. Now, as that DNA turns, it isn't going to turn nice and evenly like this. That DNA is going to turn really quick sometimes and then lag, and then turn really quick and then lag, and turn really quick and lag. And so because of that, it'll have spots where it spins a lot and spots where it doesn't spin so much. Those spots where it spins a lot and not a lot are called the major and minor grooves. Another thing that we can see in this picture of DNA that's really blown up, so we're looking really, really closely at one strand of DNA, is that you can tell that these sugars are going in this direction and these sugars are going in this direction. Because they're going in opposite directions, they're called anti-parallel. This strand goes up and this strand goes down. Alright, so we have DNA. It has two strands. It makes a twisting motion called the helix. There's 10 bases every time that helix turns, and it goes in opposite directions. We mentioned a little bit these rungs of the ladder. Let's talk about that some more. The rungs of the ladder are called bases. The rungs, each rung is made up of two different bases, and there's always different types of bases. The first type we call A, and that stands for adenine. The next one we call G, which stands for guanine. The next one is T for thymine, and the last one is C for cytosine. Now all of these bases aren't the same size. Here's how big G is. It's huge, right? And here's how big A is, also pretty big. T is a little bit smaller, and C is even smaller yet. And so what we can see, because T and C are smaller, we call them purines. P-U-R-I-N-E-S. Adenine and guanine are bigger, and so we call them pyrimidines. remember the difference is because pyrimidine is a bigger word than purine, so pyrimidine is going to describe the bigger bases. Now remember I said that bases come in pairs. There needs to be two bases right next to each other. Because we want our ladder to be nice and straight like this ladder, and not all crooked, getting bigger and smaller like this ladder, we need to have a consistent size of bases being used. And so what happens is, we'll have one really big base, like a pyrimidine, and we'll put it together with one really small base, a purine. So A and T, because one's big and one's small, will always go together. They also can go together because they have the same bonding. They're like two puzzle pieces that match up. So A and T are always going to go together. G and C, because one's big and one's small, and they have the same puzzle pieces that go together, will always go together as well. So if we go back to our picture of DNA, and we see that on one of these bases, they're just represented as rectangles here instead of nice molecules. If we have one big one here, like a G, we know G is always going to match up with C. And so if this is G, C is going to go right here. Every time G and C are together, they make two bonds. Since this big base is A, we know we have to have a small base here so we can have a consistent size, and A is always going to go with T. Now we have A, but it's over here instead of on this side, and so this side is going to have T. Right here we have a G, and it's on the right side, so on the left side we know we have to have a C, because G and C go together. On the bottom we have A over here, so we know that T is going to go over there. So always, 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 C is going to go with G, and A is going to go with T. When that doesn't happen, we have something called a mutation, and that can cause a lot of genetic disorders, which we'll talk about another time. So quick recap, there's 4 types of bases. A and G are big ones called pyrimidines. C and T are small ones called purines. Because we want to have a consistent size, we always have a big base matched up with a small base. And so A and T will always go together, and G and C will always go together. Those bases make up the rungs of the ladder. If you have any questions, as always, let me know. I'm more than willing to help.